I'm Linda Kenney, and I'm a professor at the Mechanobiology Institute at the National University of Singapore. Our work has focused for years on signal transduction in E. coli and more recently on Salmonella pathogenesis. Salmonella emerged as a pathogen when it diverged from E. coli by the acquisition of pathogenicity islands. And these two in particular encode type 3 secretion systems, which are specialized secretory systems that enable the secretion of virulence factors into the host. The acquisition of these pathogenicity islands enabled Salmonella first to colonize the intestine and later to survive in the macrophage vacuole. So we have focused on understanding the virulence factors secreted through these specialized secretory systems and how these systems are regulated. What we found is that the key regulator that is involved in activating transcription of virulence factors that enable survival in the macrophage vacuole was actually playing a dual role, that it could actually drive expression of biofilm phenotypes that we believe are essential for Salmonella to colonize and survive in forming biofilms on gallstones in the gallbladder, and that this is really the basis for the carrier state. So we see with Salmonella typhi, the great example that we all know of is Typhoid Mary, where she was colonized by Salmonella typhi, but she was asymptomatic. So she shed Salmonella in her feces for years, but she herself didn't suffer from the disease. And what we now appreciate is that this key regulator, which is sitting on a pathogenicity island, which means it was acquired during evolution, is regulating both virulence factors and pathogenesis genes, but it's also regulating ancestral genes that lead to the biofilm phenotype. And what Stuti found in this work that was novel is that this regulator could function without its kinase.
And that is what we found by AFM images, is that SsrB unphosphorylated can bring about conformational changes, and this conformational changes are sufficient for activation by binding of RNA polymerase and thereby expression of the biofilm matrix and further leading to activation of the biofilm formation in Salmonella typhi.
You can see that we are branching out now to work on both Salmonella typhi, how Salmonella maintains the carrier state, and we're also very interested in a new invasive strain of Salmonella typhimurium, which is causing high mortality rates in South Vietnamese HIV-positive patients.